So let's start with eukaryotic cells. So first up is the nucleus. The nucleus is surrounded by a double membrane called the nuclear envelope. The nucleus also contains genetic material called DNA, which are tightly wound about histone proteins and condensed into chromosomes. The function of the nucleus, as a summary, is that it is a control center for the cell. It stores organisms' genomes, it transmits genetic information and provides instructions for the protein synthesis. The nuclear envelope that surrounds the nucleus acts as a containment, so it separates the contents of the nucleus from the rest of the cell, and the pores in the nuclear envelope allow large molecules to leave, i.e. such as the mRNA. Next is the rough endoplasmic reticulum. The structure is a system of membranes containing fluid-filled cavities called cisternae, and this is continuously surrounded by ribosomes. The function of the endoplasmic reticulum is that it acts as a transport system. The cisternae act as channels that transport substances from one area of the cell to another and acts as a large surface area for ribosome reactions. The proteins, once synthesized, pass through the cisternae and are transported to the Golgi apparatus. The smooth endoplasmic reticulum has the same structure as the rough endoplasmic reticulum, except that it has no ribosomes on its outer surface. Its function is that it's responsible for the synthesis of cholesterol, lipids and phospholipids, steroid hormones, and it is also involved in the absorption, synthesis and transport of lipids in the gut. The Golgi apparatus. The Golgi apparatus, once it has received proteins, modifies them. So it can do this by adding sugar molecules to make glycoproteins, folding proteins into their 3D shapes, making lipoproteins by adding lipids, and after this, they are packed into vesicles and they are pinched off and are either moved to the plasma membrane and incorporate into it or exported, or stored in the cell, and vesicles bring materials to and from it, and it is membrane bound, which is its structure.
Next is the mitochondria. These may be spherical, rod-shaped or branched and are 2 to 5 nanometers long. They are also surrounded by two membranes with a fluid-filled space between them. The inner membrane is highly folded into cisternae, so you can see that there. Well, not very clearly. The inner part of the mitochondrion is a fluid-filled matrix. There's also an inner membrane, an outer membrane, and an intermembrane space. ATP energy currency is produced in the mitochondria during aerobic respiration, and more can be made if energy need increases through self-replication. They are abundant in cells where much metabolic activity takes place, so for example in liver cells and at synapses between neurons where a neurotransmitter is synthesized and released.
Chloroplasts. They are 4 to 10 nanometers long. They are surrounded by a double membrane or envelope. The inner membrane is a continuous stacks of flattened membrane sacs called thylakoids. Thylakoids contain the chlorophyll. Each stack of thylakoids is called a granum. The chloroplasts contain loops of DNA. This is the site of photosynthesis in plants. Stage 1 of photosynthesis, when light is trapped by chlorophyll, it is used to make ATP. This occurs in the grana. Water is also split into hydrogen ions. Stage 2, the hydrogen reduces carbon dioxide using energy from ATP to make carbohydrates, and this occurs in the stroma. Chlorophyll is most abundant on the mesophyll layer of the plant leaf.
Next we have the vacuole, lysosome, and cilia. The vacuole is surrounded by a membrane, tonoplast. It contains fluid only in plants, maintains cell stability, so when it's full of water, it pushes against the cell wall, which makes it turgid. The lysosomes are small bags formed from the Golgi apparatus, and they are surrounded by a single membrane. They contain powerful hydrolytic enzymes, which are kept separate from the rest of the cell. These lysosomes can engulf old cells and foreign matter and then recycle the components to be used within the cell. Microtubules, like cilia, protrude from the cell. Ribosomes are small spherical organelles which are 20 nanometers in size. They are made up of rRNA, ribosomal RNA. They are made in the nucleus out of two subunits which pass through the nuclear envelope into the cytoplasm and combine. Ribosomes bound to the RER are for synthesizing proteins that will be exported outside the cell. Next we have centrioles. They consist of a nucleus and a centrosome. They consist of two bundles of microtubules at right angles to each other. Microtubules are made of tubulin protein subunits and are arranged to form a cylinder. Now we have the cytoskeleton, a network of protein structures within the cytoplasm. It has a rod-like microfilaments made of subunits of the protein actin, polymers of actin, and each microfilament is about 7 nanometers in diameter. Microfilaments within the cytoplasm give support and mechanical strength, so this keeps the cell shape stable and allows for cell movement. Also, it provides cell support in the shape and helps substances and organelles to move through the cytoplasm within a cell. They form the track along which motor proteins walk and drag organelles from one part of the cell to the other.
The cell wall, the cellular cell wall. It is only present in plant cells and is absent from animal cells. This prevents the plant from bursting when it is turgid. The cell walls provide strength, support, and maintains the shape of the cell. They are permeable, and so they allow solutions passing through, whether these are solutions or solvents. It is made from a bundle of cellulose fibers, and in fungi walls it contains chitin, not cellulose. Finally, to finish, prokaryotic cells. These are very similar to eukaryotic cells in the fact that they have a cytoplasm, ribosome, and a cell wall and a plasma membrane, except they have flagella, which act like tails, which allow them to move. They have pili, which are small hair-like structures, which helps the bacteria to adhere to each other and to host cells. They have plasmids, which acts like is a small loop of DNA. Nucleoid, which is an area in the cytoplasm where DNA is positioned. A waxy protective capsule, which protects it from neighbouring cells.
Didn't realise we were listing all of them.
And just to list a few.
Keep going.
Just list a few. It does not have a nucleus, and it does not have membrane-bound organelles such as a mitochondria, Golgi apparatus, or chlorophyll. The end.
Thank God. Government! I don't know how we're supposed to do that in 2 minutes.